specific proteins. Virus symmetry. The nucleocapsids of viruses are constructed in highly symmetric ways. Symmetric refers to the way in which the protein morphological units are arranged in the virus shell. When a symmetric structure is rotated around an axis, the same form is seen again after a certain number of degrees of rotation. Two kinds of symmetry are recognized in viruses, which correspond to the two primary shapes, rod and spherical. Rod-shaped viruses have helical symmetry, and spherical viruses have isohedral symmetry. In all cases, the characteristic structure of virus is determined by the structure of the protein subunits of which it is constructed. A typical virus with helical symmetry is a tobacco mosaic virus. RNA viruses in which the 2130 identical protein subunits are arranged in the helix. The overall dimensions of the TMV virion are 18 times 300 nanometers. The lengths of a helical virus are determined by the length of the nucleic acid, but width of the helical virus particle is determined by the size and packaging of the protein subunits. The isohedron is a symmetric structure roughly spherical in shape that has 20 faces. Isohedral symmetry is the most efficient arrangement for subunits in a closed shell. Because it uses the smallest number of units to build a shell, the simplest arrangement of morphological units is 3 per face, for a total of 60 units per virus particle. The 3 units at which face can be either identical or different. Most viruses have more nucleic acid than can be packed into a shell, made of just 60 morphological units. The next possible structure that permits close packaging contains 180 units, and many viruses have shells with the configuration. Other known configurations involve 240 units and 420 units. A model of an isohedral shows an electron micrograph of a typical isohedral virus. Human papilloma virus shows a computer model of the same virus. Envelope viruses. Many viruses have complex membranes, structures surrounded by the nucleocapsid. Envelope viruses are commonly in an animal world. Influenza virus, but envelope bacteria and plant viruses are also known. The virus envelope consists of a lipid biolayer with proteins, usually glycoproteins, embedded in it. The lipids of the membrane are derived from the membranes of the host cell, but the proteins are encoded by the virus. The symmetry of envelope viruses is expressed not in terms of a virion as a whole, but in terms of nucleocapsid present inside the virus membrane. What is the function of the membrane in a virus particle? We will discuss this in detail later, but note that the membranes in the structure component of a virus particle that interacts first with cell. The specificity of a virus infection and some aspect of virus penetration are controlled in part of the characteristic of a virus membrane. Complex viruses. Some virions are even more complex, being composed of several separate parts, each with separate shapes and symmetries. The most complicated viruses in terms of structure are some of the bacterial viruses, which possess not only isohedral heads but also helical tails. In some bacteria viruses, such as the T4 virus and other Escherichia coli, the tail itself has a complex structure. For instance, T4 has almost 20 different proteins in the tail, and the T4 head has several more proteins. In such complex viruses, assembly is also complex. For instance, in T4, the complete tail is formed as a subassembly, and then the tail is added to the DNA-containing head. Finally, tail fibers formed from another protein are added to make the mature infectious virus particle. Enzymes in virions. We have stated that virions do not carry out metabolic processes outside a host cell. A virion is metabolically inert. However, some virions do not contain enzymes that play a role in the infection process. Some of their enzymes are required for very early events in the infection process. Virions infecting some bacteria possess an enzyme, lysozyme, that makes a small hole in the bacteria cell that allows the viral nucleic acid to enter. Lysosome is produced in large amounts in the later stages of infection, causing lysis of the host cell and release of the virions. Also, some viruses contain their own nucleic acid polymerase that transcribe the viral nucleic acid into messenger RNA without using the cellular enzymes, or because they require an enzyme the cell does not have. For example, retroviruses are RNA viruses that replicate inside the cell as DNA intermediates. These viruses possess an enzyme, an RNA-dependent DNA polymerase called reverse transcriptase, that transcribes the information in the incoming RNA into DNA intermediate. Other viruses contain enzymes that aid in release of the virus from the host cells in which they were produced. One group of such enzymes, called neuramindases, break down glycosidic bonds of glycoproteins and glycolipids of the connective tissue of animal cells, thus aiding in the liberation of the virus. Growth and quantification. Because viruses replicate, only living cell research on viruses require use of appropriate host of the three types of host: bacteria, plants, and animals. Viruses infecting bacteria are typically the easiest to grow in the laboratory. For the study of bacteria viruses, pure cultures are used either in liquid or semi-solid, agar, media because many bacteria are so easy to culture. It is quite easy to study bacteria viruses, and this is why such detailed knowledge of bacteria viruses multiplication is available. Plant viruses can be more difficult to work with. Since their studies sometimes require use of the whole plant, plant viruses also often require a break in the thick plant cell wall in order to infect. Sometimes these viruses are assayed by mechanically damaging a plant leaf and applying virus to the damaged area. However, some plant viruses can be studied using protoplast or other cell culture techniques. Although initially an animal virus may be grown in a whole animal that is susceptible to the virus, for research purposes, a more manageable host is desirable. Fortunately, many animal viruses can be cultivated in tissue or cell cultures, and the use of such cultures has enormously facilitated research on animal viruses. A cell culture is obtained by promoting growth of cell taken from an organ of the experimental animal. Cell cultures are generally obtained by aseptically removing pieces of tissue in question, disassociating the cells by treatment with an enzyme that breaks apart the intercellular cement, and spreading the resulting suspension out on the bottom of the flat surface, such as the bottle or Petri dish. The cells generally produce glycoprotein-like materials that permit them to adhere to glass surfaces. The thin layer of cell adhering to glass or plastic dish, called a monolayer, is overlaid with a suitable culture medium and the culture incubated. The culture media used for cell cultures are generally quite complex, employing a number of amino acids and vitamin salts, glucose, and bicarbonate buffer system. To obtain but growth addition of small amount of blood serum is usually necessary, and several antibiotics generally aided to prevent bacteria contamination. A colored pH indicator is added to help detect this contamination. Some cell cultures prepared in this way grow indefinitely and can be established in permanent cell lines. Such cell cultures are most convenient for virus research because cell material is continuously available for research purposes. In other cases, indefinite growth does not occur, but the culture may remain alive for a number of days. Such cultures, called primary cell cultures, may still be useful for virus research, although new cultures will have to be prepared from fresh sources from time to time. In some cases, cell culture monolayers cannot be obtained, but whole organs or pieces of organs can be cultured. Such organ cultures may still be useful in virus research because they permit growth of virus under more or less controlled laboratory conditions. There is great specificity between the virus and the host than simply determining to which kingdom or phylum the host belongs. In some cases, the specificity of extreme, the virus may only be able to infect and or multiply in a single species or subspecies, and sometimes only some tissues within the host. In other cases, however, the specificity is broader. We will discuss some of the reasons for this specificity in the following sections. Quantification of viruses. Quantification of viruses. In order to obtain any significant understanding of the nature of viruses and virus replication, it is necessary to be able to quantify the number of virus particles. Virions are almost always too small to be seen under the light microscope, although they can be observed under the electron microscope. The preparation of samples for observation can be too cumbersome to use simply for quantification. In general, viruses are quantified by measuring their effects on their host cells that they infect. In this context, a virus infection unit is the smallest unit that causes a detectable effect when placed with susceptible host. By determining the number of infectious units per volume of fluid, a measure of viruses quantified can be obtained. We discuss here several approaches to assessment of virus infection unit, limiting our discussion to bacteria and animal viruses. Plaque assay. When the virion initiates an infection on a layer of lawn host cells grown on a flat surface, a zone lysis or zone of growth inhibition may occur that results in a clear area of the lawn of growing host cells. This clearing is called a plaque, and it is assumed that each plaque has originated from replication, even that began with one virion. Plaques are essentially windows in the lawn of confluent cell growth. With bacteria viruses, plaques may be obtained when the virus particles are mixed into a thin layer of host bacteria that is spread out as the agar overlay on the surface of the agar medium. During incubation of the culture, the bacteria grow and form a turbid layer that is visible to the naked eye. However, wherever a successful viral infection has been initiated, lysis of the cells occurs, resulting in the formation of plaque. The plaque produced also permits the isolation of pure virus stains because of